Welcome all, and thank you for joining today's AWRI webinar. My name is Ben Cordingley, and I'm an enologist at the AWRI. In today's session, we'll be hearing about the use of non-Saccharomyces yeast in winemaking. In the spirit of reconciliation, the AWRI acknowledges the traditional custodians of country throughout Australia and their connections to land, sea, and community. We pay our respect to their elders past and present, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. I would also like to acknowledge Wine Australia for providing funding and support for webinars via the AWRI Extension Project. Before we jump in, a couple of reminders for anyone who is new to AWRI webinars. If you'd like to provide a comment or ask a question, please click on the Q&A button on the Zoom toolbar, type in your question and click send to shoot it through. We'll be holding the Q&A session at the end of this presentation, but feel free to send through your questions at any stage. A reminder that this session is being recorded and a link will be sent through to you later to view on the AWRI's YouTube channel. We'd also like to collect data today on your knowledge of this topic. You should see a one-question survey pop up on your screen asking you to rate how much you know about using non-Saccharomyces yeast in winemaking. If you could please rate your knowledge on this topic on a scale between 0 to 10 now, with 0 being nothing to 10 being expert or black belt level. At the conclusion of today's webinar and after the Q&A, we'll prompt you again with this question to establish your knowledge of the topic after the webinar. Again, you'll be asked to rate your knowledge on the scale of between 0 to 10. For anyone who has just joined, welcome. Today's topic is the use of non-Saccharomyces yeast in winemaking. It is with great pleasure that we welcome Dr. Anthony Borneman from the Australian Wine Research Institute today. Anthony is the Molecular Biology Research Manager at the AWRI and an affiliate of the University of Adelaide. Anthony leads research that is focused on applying genomics to understand the genetic basis of biological diversity in winemaking, including grapevines, commercial wine yeast and bacteria, and wild ferments. Anthony, if you're ready to make a start, I'll hand over to you.
Thanks, Ben. Let's get my screen up. Hopefully you can see the screen okay.
Yeah, perfect, Anthony.
Thanks. So I'll just spend a little bit of time going through non-Saccharomyces yeast in winemaking, both from a wild ferment perspective and from the use of commercial non-Saccharomyces starters. So first off, sort of where do all these yeasts come from that we see or encounter in winemaking? And the fact is they come from everywhere in the environment, from the soil in the vineyard, from on the bark, the vine itself, also growing on the outside of the grapes. On that wax sort of cuticle of the grapes, you get a lot of yeast that live there year in, year out on nice undamaged fruit. And of course, when grapes are also damaged, we get a whole another set of yeast strains that can come in. And then there's also yeast that sort of appear, or at least appear to live primarily in the cellar and come through at the fermentation stage. When it comes down to what variables can affect yeast populations, pretty much anything you can do as far as winemaking interventions will affect yeast populations, and also, just basic environmental parameters: how much rain, humidity, the temperature that's around, agrochem use, proximity to other orchards or vineyards, and then some of the others that I've mentioned, sort of berry damage, whether you're hand harvesting versus machine harvesting, because machine harvesters can act as almost an inoculation source for yeast, which sort of marries in with harvester hygiene, whether or not you're using SO2 and how much you're using, whether you're transporting your fruit, and then just other winemaking basics around whether or not you're using solids, what level of solids, whole berries or bunches, cold soaks, oxygen exposure. So many, many things. I won't be able to touch on all these today. I will touch on some of the winemaking interventions you can get into to sort of try and tailor yeast populations, but certainly pretty much anything will touch on or influence the yeast that you see in your ferments. So when you're winemaking, it's not just Saccharomyces. There are a great many different yeast in there, and the populations change over time and also change depending on what species. So, for example, for your non-Saccharomyces yeast at the start of fermentation, you'll have a certain level, the cell concentration of your non-Saccharomyces yeast, and as the ferment progresses, these will go up in concentration a little bit and then die off. And the main thing that causes your non-Saccharomyces to die off is Saccharomyces cerevisiae, so the major wine yeast coming through. So generally on clean fruit, and this is in an uninoculated situation, your Saccharomyces will be at a lower proportion than your non-Saccharomyces yeast. But as the ferment kicks off, Saccharomyces will quickly take over, starts converting a lot of sugars into alcohol, and it's that action of Saccharomyces that actually contributes to the pretty rapid decline of your non-Saccharomyces yeast. Now, in this situation, if you were to inoculate with Saccharomyces, then your levels of Saccharomyces would come in much higher than your non-Sacs at the inoculation point, grow up, reach a big level before they start to tail off. But importantly, because you're inoculating with a large amount, your non-Saccharomyces will actually dive down much, much quicker and play a much smaller role in influencing the fermentation outcomes. Now, hopefully you don't see Brettanomyces come through. Brettanomyces is generally at ferments in almost undetectable levels, and then will jump up at the end of primary, and often during barrel storage, although there is anecdotal evidence for Brettanomyces coming up in ferments that haven't seen oak as well. And then, of course, Enococcus, which is kicking around during ferment, survives primary ferment to then go through MLF once primary ferment's over. So there's this sort of dynamic in yeast populations and bacteria with Enococcus listed here that occur during fermentation, with different populations sort of going through increases and decreases in their cell concentrations, and they're all dependent on each other. So we've now concluded a very large project around wild fermentation or uninoculated fermentations, looking at which species are involved in wild fermentations around Australia and also the progression during ferment. This is just a snapshot, a sort of averaged out look at a wine ferment based on hundreds of wild ferments that we've looked at over the course of 5 years in the project. So we've probably looked at upwards of 1,000 ferments from around Australia, both red and white, different varieties, but mainly focused around Chardonnay, Shiraz, and Pinot. And so these are 4 different time points that we've looked at, going from just after the grapes are crushed through to the end of ferment, with the bars representing the abundance of particular genera of yeast and yeast-like fungi that we've been looking at using a DNA sequencing approach to quantify their presence in the ferment. So each colour is a different genus. Lots of colours. It is hard to tell them apart once you get to these colours, but I'll point out a few key ones. And again, this is at the genus level. So there can be many species that contribute to each of these colours within a particular genus. So at the start of crush, lots and lots of different species. Very diverse. Some ferments can have upwards of 50 or 60 species in there. Big contributor is these species that live on the berry all of the time, whether it's damaged or healthy, such as Oribasidium in the dark blue, Cladosporium in the brown. And then some of the other non-sacs you may be more familiar with, such as Hanseníaspera, Pichia, Meschnickauer. This light blue that we see jump through, this is Saccharomyces, so it starts off as a very minor proportion at crush. And in fact, healthy, intact grape berries, you pretty much don't find Saccharomyces. It is not able to survive on the outside of a healthy berry. It's only once you've got some damage in there and some sugar leaking out that Saccharomyces can actually be an inhabitant of the grape. And then as ferment goes through, a lot of these sort of inhabitants of the grape cuticle die off. So we see the Oribasidium, Cladosporium start to go down their proportions. Some of these other non-sacs have their little time in the sun, such as Hanseníaspera, Meschnickauer, before Saccharomyces really takes over. So that's an average sort of ferment. But what we can do with the data is sort of break it down by individual wineries, for example. So this is results for a 2021 wild fermented Chardonnays. And these are pairs of ferments from different blocks within individual wineries, and again, color-coded. But hopefully you can take in, you can't tell all the detail, but there are very different colors for some of the wineries. So you have quite a few ferments that are dominated by the Hanseatic aspera, this goldy color during ferment. But then you have others where there's pinks and reds and greens, so candidas and Isachenkia, pickias that are coming in. So there is variation between wineries, across wineries, across regions, but also within the same winery where you get in fruit from different locations. This can have very big differences between the wild yeast populations that are present in the ferment and how the ferment progresses. So what do we see as far as wine effects from wild fermentation and the presence of these non-Saccharomyces yeasts for extended periods of time? So as far as trends go, you do see that wild ferments can have higher residual sugars, often contain less alcohol, often a bit higher TA, but also higher glycerol, lower SO2, lower acetaldehyde, and you can see a little bit of higher VA and lower tannins. You also see from a sensory standpoint that wild ferment wines are more diverse and can be more complex, whereas inoculated wines are generally more uniform, dominated by acetate esters. So wild ferments can give you much more complexity, much more diversity, but there are also downsides and risks involved, mainly around VA. So as far as wild ferments go, it can be a case in a very minimal intervention standpoint of crushing your grapes, letting them go. But there are winemaking interventions that can and do shape yeast composition in a wild ferment format, such as the use of SO2 prior to fermentation, aeration, temperature, many others that I mentioned before, but I'm going to concentrate on these three and just sort of outline some of the effects we see on wild ferment populations and non-sac populations with these winemaking interventions. So as far as SO2 addition goes, this can have some pretty major effects on wild fermentation. A lot of the non-Saccharomyces yeasts are very, very sensitive to SO2. And so this was a study we did, and this has been repeated now with many commercial Chardonnays from this particular winery. This is from 2019. And we dosed up with either 0, 10, 20 or 40 parts of SO2 just prior to fermentation. And there had been no SO2 added at the winery or at the crusher or anything like that. So this was total SO2 added just prior to ferment. And what we see if we look at sort of genus level here on the left plots is that all the ferments sort of gave us the trend we were looking for with Saccharomyces coming up as the ferment went through. They were very much dominated by Hanseatic aspera as the ferment went on as well. So this purple with Saccharomyces in blue. So this is T1 through T4, so crush through the end of ferment, and then the SO2. What we saw with the use of the SO2, though, was that the actual contribution of Hanseatic aspera went down, and we saw some of this candida jump up. So even at the genus level, we're seeing effects based on SO2. But when we narrow it down to the species level, and in fact, Hanseatic aspera is one of these ones, a genus where there are at least 4 or 5 different species that can be involved in the wine fermentation, we see that we get a shift in the use of SO2. So at about 25, 30 parts SO2, we see a complete shift in the species from this maroon one, which is our Hanseatic aspera uvarum, through to this pink one, which is Hanseatic aspera osmophila. And when we look at the chemical analysis of these wines, where we've added the SO2, we actually see that as we've gone up in our SO2 use, we're getting a lot more of these compounds such as phenylethyl acetate, some of our other fatty acid esters, apple characters from ethyl hexanoate, and a lot of these sort of solvent characteristics going down. So actually the use of the SO2 is pushing the populations in different directions and giving us changes in the chemical composition, which are predicted to be very sensorially relevant. This is sort of 10 times as much to phenylethyl acetate and well outside what we, the odor threshold, well above that. So the use of SO2 can sort of push ferments one way or the other from a sensory standpoint, wild ferments. Aeration is another intervention you can use on wild ferments. We've looked at both the timing of aeration, so whether we put air in after 24, 48 or 72 hours from the start of fermentation, or aeration intensity at 24 hours, low, medium and high. Take homes here is that early addition of air stimulates the non-Saccharomyces yeast. Later addition of air stimulates Saccharomyces, so wild Saccharomyces, and can, in all situations, the addition of air makes the ferment go through much faster and in a more robust way. You do have to be careful though, especially in whites, in that once we get up into these sort of medium and high aeration intensities, you do start to see enhanced production of those sort of acetic VA compounds, such as ethyl acetic acid up here with our high air, correlating with increased risk of the production of acetic acid. But certainly for robustness, so not a lot of differences in flavour apart from that risk of VA, but certainly from a ferment robustness standpoint and getting ferments through, wild ferments through much faster and more reliably, a little bit of air can go a long way to helping you out there. So I'll leave wild ferments behind now and sort of have a look and finish up on non-Saccharomyces starter cultures, because of course not everyone wants to do a wild ferment to get the advantages of non-Saccharomyces characteristics into their ferments. So these provide an alternative to spontaneous fermentations. They can help provide those novel or complex flavour profiles without having to go down the wild ferment path. There are quite a few non-Saccharomyces commercial starter yeast cultures around, or mixtures around, so well over 20 now and growing all the time, with a lot of different applications. So for everything from reducing alcohol through deacidification, aroma, mouthfeel, bioprotection, flavour complexity, and also acidifying now with the use of some of the non-sacs such as Lachantia. So as far as what all these non-sacs do, they do tend to have different and overlapping roles, and the use of single species of non-sacs can provide more than one sort of characteristic to your ferment. Some are very defined, such as bioacidification that's promoted by Lachantia, or bioprotection from species such as Meshnikaria fructicola, which itself doesn't undergo, or usually is not seen in wine fermentation and doesn't sort of contribute to wine ferment, but can be used as bioprotection. So you've got the production of esters or thiols or terpenes, and then many of these species will do one or more of these characteristics. Looking at some in isolation, for example, so for Toleraspora, it's often seen to bring out sort of tropical fruit and varietal characters, overall impressions compared to a straight Cerevisiae. And these are used as sort of sequential or co-inoculations. And then again, with Lachantia, adding to or performing that bioacidification role. So as far as where we get these non-Saccharomyces and, what sort of process we go through to get these for use in the wine industry, we've been doing some work at the AWRI now, putting non-Saccharomyces, or a developmental process for non-Saccharomyces yeast. We've, over the years, isolated thousands of Saccharomyces and non-Saccharomyces cultures from wines. We've been going through a large-scale screening process now where Christian Varela's actually looked at well over 1,000 individual wine yeast isolates, and he's been screening these for winemaking relevant attributes, everything from their ethanol tolerance, whether they can survive in really high sugar, if they're resistant to the presence of copper, can they grow in anaerobic environments that are present in ferments, same with low temperature, are they SO2 resistant, do they make H2S, which is a negative, of course, and then a lot of different enzyme activities, whether they express these enzyme activities, because often these enzyme activities directly impact on non-volatile grape-derived flavour precursors to release the volatile component and add flavour and aroma aspects to wine. We concentrated on a couple of these enzyme activities, beta-galactosidase and beta-lyase. So beta-galactosidase for thiols release, for monoterpene release, beta-lyase for thiol release, and sort of got 20 or so isolates that we've been screening through. We've settled on a few strains that got through, that we've taken through to winery scale trials, a Hansen Aspera Yavarum. And this is a strain, Hansen Aspera is one of those common ones I mentioned before from a wild ferment standpoint, of which so far there's not much in the way of commercial strains, although there are some now in the pipeline. Mesonicaria pulcherrima that express beta-lyase for high thiol production, and a Toleraspora that come through with, which we saw some enhanced thiols and ethyl esters during the screening. So as far as taking these through, we've now taken them through to sort of pilot winery scale fermentation, because you start off small. All that screening was done on plates, thousands of strains, little tiny ferments, and then we've finally gone from 1,200 down to 3, which we've taken up for pilot wide scale ferments and sensory. And we see when we put these through, and these again are co-inoculated or sequentially inoculated with Saccharomyces, compared to the Saccharomyces control, we are seeing sensory differences with our Mesonicaria and Hansen Asperas down here on the PCA in this sort of tropical fruit area. From a chemical standpoint, the Hansen Aspera did release more monoterpenes, while the Mesonicaria did release those thiols that we thought it would. And the Toleraspora up here with sort of stone fruit, citrus from those esters and thiol release. So these are all scored favourably compared to the straight Cerevisiae control. So they do add flavour and aroma complexity. So now we're looking to do more studies on these, and then hopefully they'll come out as commercial yeast strains sometime in the future. So I'll just finish off there. Very quick from a very big area and a very growing area with non-Saccharomyces. So the take-home message is, there are hundreds potentially of non-Saccharomyces species that we observe in wine, and each will have their own influences and pros and cons when we get into winemaking. And these do vary based on winery, vintage, and region. So if you are going down the use of non-Saccharomyces via a wild ferment situation, your wines will be, or your populations will be unique both to your winery, your area, your year, and they do change over time. So it does take a bit of work to get used to dealing in a wild ferment sort of matrix from year to year. And interventions are very important as far as they will change your microbial composition, and they can shift flavour diversity, especially in the use of SO2. Aeration is more an efficiency, robustness thing, but again, if you're worried that wild ferments won't go through, a little bit of air at the right time can actually help get your wild ferments and your wild yeast to finish the job. For those that don't want to go down the wild ferment path, though, there are many non-Saccharomyces starter cultures that are commercially available, more and more each year, and they can be used to increase colour, aroma, complexity, mouthfeel, and also from a bioprotection standpoint, protect from spoilage pre and post fermentation. So they are worth looking into. Whether or not you're also looking at non-sac from a wild ferment standpoint, having those available for inoculated ferments can be really useful, or in years when you don't want to go down the wild ferment path. So I'll finish off there just with acknowledgements. Wine Australia supporting the work, and also many people from AWRI for strain ID, helping with all the screening. Christian Varela, who did a lot of the work, also in the screening part, and Yolumba with Greg and Louisa for helping out with winery scale pilot ferments. And again, also a lot of the wineries that helped us out with the wild ferment project that's now come to an end, looking at those yeast populations across wild fermentation. So I'll finish off there. Happy to take questions. Yeah, thank you.
Thanks very much, Anthony. So send your questions through, and we'll put those through to Anthony. So I've got a question here, Anthony. Lachrantia thermotolerance. So how dramatic can that bioacidification be? And is it possible to stop it once you've reached the desired level of acidification?
Yep. There are a couple of different strains available that have slightly different impacts, but it can be quite dramatic. I think it can drop it depending on how long you want to leave it in your juice for too, and the amount, 0.4, 0.5 pH. The way to stop it, from what I understand from the companies that sell it, is you time when you seed over the top with Saccharomyces. So it's an amount of non-sac you add, and how long you hold off seeding over the top with a Saccharomyces strain to give it a chance of your time to do its thing.
Great, thanks, Anthony. Another question has come through. Could you please advise the rates for low, medium, and high O2 additions?
Yeah. So I'd have to grab that out, the specific numbers. It was pretty much saturated, as much saturation as we could get for 24 hours at each of those time points. So that may be a bit harder to achieve in a winery situation because we were dealing with smaller size ferments. But essentially we were getting, it wasn't micro-ox. We were saturating oxygen for those sort of 24. Low, medium, and high, I'd have to get those numbers out. That is published now, so we can push that paper around for people that are interested in the exact numbers, but I haven't got them off the top of my head.
Great, thanks. For the person that asked that question, if you want to send us an email at the AWRI help desk, we can get that paper out to you, as well as the details of those O2 rates.
Yep.
Another question here, Anthony. Has there been any studies done on the impact of non-sac yeasts and the consumption of vitamins, minerals, nitrogen versus Saccharomyces strains?
Yep. So they do. So you do have to be careful from the standpoint of using non-sacs. They all use different vitamins, different amounts of nitrogen, different nitrogen sources. So some will not play nicely with Saccharomyces. So that's one of the important things when developing these strains, is developing strains that you can seed Saccharomyces in over the top of. Certainly in a wild ferment situation, if your fruit is too clean and you don't have Saccharomyces in there, if you get too many non-sacs, it can mean that your ferment doesn't go through if Saccharomyces can't get a hold. So yes, they do. They can tie up vitamins, minerals, nitrogen sources away from Saccharomyces, but usually in most ferment situations, they'll die off and release those vitamins back into the ferment.
Thanks, Anthony. We have a question here from Mary, who's asked, which strains are best for lowering alcohol level?
That's a good question, because the alcohol lowering can often be it's not necessarily an every time situation. Some of the strains can and cannot, depending on the juice and other variables, lower alcohol by different amounts. Certainly they say the Lachrantia strains will lower alcohol as they're pushing carbon into acid. There's documented work out there on the Meshnikawa strains being able to, in certain juices, lower alcohol, sort of 0.5 to 1%. Again, I'd have to, for specific commercial strains, I'd have to, consult some commercial guys.
Thanks, Anthony. We haven't had any more questions come through. We have had one last minute question. So how effective is bioprotection from Mechnicovia, and will it suppress Saccharomyces or just undesirable strains, e.g. Brett/Candida?
Yeah, I mean, the bioprotection for these strains is usually for cold soak and things like that. So it is protecting from generally a lot of the other non-Sacs and things like acetic acid bacteria. And that's at those cold temperatures. So Saccharomyces normally wouldn't be doing much anyway. Usually if Saccharomyces is in there, it's going to come over the top. And that's the case with the bioprotection strains, because you will inoculate over the top of Saccharomyces and they'll take care of it pretty easy. Often one of the limitations with all of these non-Sacs, even the commercial ones, is the level of Saccharomyces that you have in your ferments. If you've got a fair bit of even wild Saccharomyces, it'll dominate a lot of these non-Saccharomyces yeast, no matter what. It does what it does. So bioprotection wise, it's more about undesirables.
Yeah, some of these bioprotection strains, Anthony, they're used at quite low rates. Is that correct?
Generally, for the bioprotection, they're actually fairly high rates. But like the Mechnicovia fructicola isn't a yeast that's involved with wine fermentation. It doesn't tend to proliferate in. So as soon as you sort of get your ferment up to temperature, it doesn't really go any further. It takes up the space during cold soak, during storage, but as far as I know, it doesn't involve itself in fermentation. It's not a wine-derived species.
And are these strains producing compounds that are stopping other things from growing, or what's the mechanism that they're working by to provide that bioprotection?
They're often just soaking up available space. So because you're inoculating at a fairly high rate, it's almost like a population crowding thing, that you just take up space away from these other species. So they do help to, they do make some compounds that help inhibit other species. It's a competitive environment. So you add a lot of these bioprotection strains, they help suppress the other undesirables. But in the case, especially of fructicola, it doesn't go any further once you get ferment up and running. So it doesn't impact the fermentation afterwards.
Great. Thanks very much, Anthony. So it doesn't look like we've got any more questions. So I'd like to thank Dr. Anthony Bornemann for joining us today and providing insights into the use of non-Saccharomyces yeast in winemaking. I'm sure everyone that attended the session today got a lot out of it. And I'd like to thank you, the audience, for logging in and taking the time to get involved. I'd like to remind you that, as always, you will receive a link to view the recording of this presentation on the AWRI's YouTube channel. So we'll be back next week with another AWRI webinar on Thursday, the 23rd of November. This will be titled The Seasonal Outlook for the 2023-24 Summer and an update on major agricultural projects. If you'd like to register for this session, please visit the AWRI website. Thanks again, and I look forward to seeing you at the next AWRI webinar.